Cycadales is an order of seed plants that includes all the extant cycads. These plants typically have a stout and woody, ligneous trunk with a crown of large, hard and stiff, evergreen leaves. They usually have pinnate leaves. The individual plants are either all male or all female. Dioecious cycads vary in size from having trunks only a few centimeters to several meters tall. They typically grow very slowly and live very long, with some specimens known to be as much as 1,000 years old. Because of the superficial resemblance, they are sometimes confused with and mistaken for palms of ferns, but are only distantly related to either. Cycadales are found across much of the subtropical and tropical parts of the world. They are found in South and Central America, where the greatest diversity occurs. Mexico, the Antilles, southeastern United States, Australia, Melanesia, Micronesia, Japan, China, Southeast Asia, India, Sri Lanka, Madagascar, and southern and tropical Africa, where at least 65 species occur. Some can survive in harsh semi-desert climates, xerophytic, others in wet rainforest conditions, and some in both. Some can grow in sand or even on rock. Some in oxygen-poor, swampy, bog-like soils rich in organic material, and some in both. Some are able to grow in full sun, some in full shade, and some in both. Some are salt-tolerant, halophytes. Cycadales belong to the biological division Cycadiphyta along with the fossil order Melanesia. The three extant families of Cycadales are Cycadaceae, Standardriaceae, and Zamiaceae, though they are a minor component of the plant kingdom today. During the Jurassic period, they were extremely common. They have changed little since the Jurassic, compared to some major evolutionary changes in other plant divisions. Cycads are gymnosperms, naked-seeded, meaning their unfertilized seeds are open to the air to be directly fertilized by pollination, as contrasted with angiosperms, which have enclosed seeds with more complex fertilization arrangements. Cycads have very specialized pollinators, usually a specific species of beetle. They have been reported to fix nitrogen in association with a cyanobacterium living in the roots. These blue-green algae produce a neurotoxin called BMAE that is found in the seeds of cycads. This neurotoxin may enter a human food chain, as the cycad seeds may be eaten directly as a source of flour by humans or by wild or feral animals such as bats, and humans may eat these animals. It is hypothesized that this is a source of some neurological diseases in humans. Description: cycads have a cylindrical trunk which usually does not branch. Leaves grow directly from the trunk, and typically fall when older, leaving a crown of leaves at the top. The leaves grow in a rosette form, with new foliage emerging from the top and center of the crown. The trunk may be buried, so the leaves appear to be emerging from the ground and the plant appears to be a basal rosette. The leaves are generally large in proportion to the trunk size, and sometimes even larger than the trunk. The leaves are pinnate, in the form of bird feathers, pinnae, with a central leaf stalk from which parallel ribs emerge from each side of the stalk, perpendicular to it. The leaves are typically either compound. The leaf stalk has leaflets emerging from it as ribs, or have edges, margins, so deeply cut, incised, so as to appear compound. Some species have leaves that are bipinnate, which means the leaflets growing along the rachis each have their own subleaflets growing along a rachia. Self-similar geometry. Taxonomy. About 306 species of living cycadales have been described, in 10 to 12 genera and 2 or 3 families of cycads, depending on taxonomic viewpoint. The classification below is based upon a hierarchical structure based on cladistic analyses of morphological, anatomical, karyological, Physiological and phytochemical data. The number of species in the clade is low compared to the number in most other plant phyla. However, paleobotanical and molecular research indicates that the diversity was greater in the history of the phylum. The disparity in molecular sequences is very high between the three main lineages of cycads, implying that genetic diversity in the clade was once high. Some clearly defined cycad species can interbreed and produce fertile offspring. Suborder Cycadinae. Family Cycadaceae. Subfamily Cycadoideae. Cycas. About 105 species in the Old World from Africa east to southern Japan, Australia and the western Pacific Ocean islands. Type C. Cercinalis L. See also C. Pruinosa and C. Revoluta. Suborder Zamianae. Family Stangeriaceae. Subfamily Stangerioideae. Stangeria. One species in southern Africa. Type S. Eriopus. Kunza. Balin. Subfamily Boaneroideae. Bowenia. Two species in Queensland, Australia. Type B. Spectabilis hook. X hook. F. Family Zamiaceae. Subfamily Encephalatoideae. Tribe Dioecious. Dio1. 13 species in Mexico and Central America. Type D. Edule Lindley. Tribe Encephalatae. Subtribe Encephalatini. Encephalatos. About 66 species in Southeast Africa. Type E. Friderici Guglielmi Lehmann. E. Transvenosis. Modjadji Cycad. Subtribe Macrozamianae. Macrozamia. About 41 species in Australia. Type M. Reedley. Fisher X. Gaudicho. C. A. Gardner. Lepidozamia. Two species in eastern Australia. Type L. Perovskiana Regal. Subfamily Zamioideae. Tribe Ceratozamiae. Ceratozamia. 26 species in southern Mexico and Central America. Type C. Mexicana Brongan. Tribe Zamiae. Subtribe Microcycadinae. Microcycas. One species in Cuba. Type M. Calicoma. Mikel. A. D.C. Subtribe Zamianae. Chigua. Two species in Colombia. Type C. Restrepoii. Stevenson. Zamia. About 65 species in the New World from Georgia, USA south to Bolivia. Type Z. Pumila L. See also Z. Furfuracea. Identification. The three families can be identified by looking at the central stalk of the leaf. Each family has at least one vein running up the leaf stalk from bottom to top. Longitudinal. The Cycadaceae have only one vein in the center of the leaf stalk. Central vein. But no veins on the stalklets of the leaflet. No lateral veins. The Stangeriaceae have only one central vein, but with lateral veins. Also, the Zamiaceae have more than one central vein, and they are parallel to each other. History of human use. Starch-rich cycad seeds were an important part of the diet of Aboriginal people in some regions of Australia. Prior to eating them, Aboriginal people used techniques such as roasting the seeds and then soaking the kernels for many days to wash out the poison. Modern knowledge about cycads began in the 9th century with the recording by two Arab naturalists that the genus Cycas was used as a source of flour in India. Later, in the 16th century, Antonio Pigafetta, Fernão Lopes de Castaneda and Francis Drake found cycas plants in the Moluccas, where the seeds were eaten. The first report of cycads in the New World was by Giovanni Lirio in his 1576 trip to Brazil, where he observed a plant named iris by the indigenous people. This species is now classified in the genus Zamia. In January 1697, Dutch sailors landed on the banks of the Swan River in Western Australia. Part of the crew of the Naptang one of the ships in an expedition led by the Dutch navigator Willem de Vlaming. They noticed palm-like plants with cones bearing large brightly colored seeds. These were enticing and appeared to be a delicious change to a monotonous seafaring diet. Some of the sailors eagerly sampled the seeds and later wrote about the experience. "I ate 5 or 6 seeds, and drank some of the water from one of the aforesaid holes. But after an interval of about 3 hours I and 5 more of the others who had also eaten the said fruit began to vomit so violently that there was hardly any distinction between death and us." Quote, in August 1770, some British sailors passed the time on the banks of the Endeavour River in northern Queensland. They were part of the crew of the Endeavour Voyage of James Cook, and the ship has been badly damaged on a coral reef. The sailors noticed many palm-like plants growing nearby, some of which had large brownish seeds described as nuts about the size of a large chestnut and rounder, thinking that the seeds were edible. Some sailors tried them. A few hours later, these men suffered from painful stomach cramps and explosive vomiting. The ship's captain later wrote in his journal, "Those who made the experiment paid dear for their knowledge of the contrary, for they, the seeds, operated as an emetic and cathartic with great violence." Pigs were taken on board the Endeavour in Tahiti as a source of fresh meat, and when they were fed cycad seeds, several of them died. Throughout the 18th–19th centuries, discoveries of several species were reported by numerous naturalist researchers and discoverers traveling throughout the world. One of the most notable researchers of cycads was American botanist C.J. Chamberlain, whose work is noteworthy for the quantity of data and the novelty of his approach to studying cycads. His 15 years of travel throughout Africa, the Americas and Australia to observe cycads in their natural habitat resulted in his 1919 publication of The Living Cycads, which remains current in its synthesis of taxonomy, morphology and reproductive biology of cycads, most of which was obtained from his original research. His 1940s monograph on the Cycadales was never published. Historical diversity. The probable former range of cycads can be inferred from their global distribution. For example, the family Stangeriaceae only contains 3 extant species in Africa and Australia. Diverse fossils of this family have been dated to 135 Maya, indicating that diversity may have been much greater before the Jurassic and late Triassic mass extinction events. However, the cycad fossil record is generally poor and little can be deduced about the effects of each mass extinction event on their diversity. Instead, correlations can be made between the number of extant gymnosperms and angiosperms. It is likely that cycad diversity was affected more by the great angiosperm radiation in the mid-Cretaceous than by extinctions. Very slow cambial growth was first used to define cycads, and because of this characteristic the group could not compete with the rapidly growing, relatively short-lived angiosperms, which now number over 250,000 species, compared to the 947 remaining gymnosperms. Origins. The cycad fossil record dates to the early Permian, 280 million years ago. Maya. There is controversy over older cycad fossils that date to the late Carboniferous period, 300 to 325 Maya. One of the first colonizers of terrestrial habitats. This clade probably diversified extensively within its first few million years, although the extent to which it radiated is unknown because relatively few fossil specimens have been found. The regions to which cycads are restricted probably indicate their former distribution in the Pangaea before the supercontinents Laurasia and Gondwana separated. Recent studies have indicated the common perception of existing cycad species as living fossils is largely misplaced. with only Bowenia dating to the Cretaceous or earlier. Although the Cycad lineage itself is ancient, most extant species have evolved in the last 12 million years. The family Stangeriaceae, named for Dr. William Stanger, 1811–1854, consisting of only 3 extant species, is thought to be of Gondwanan origin, as fossils have been found in Lower Cretaceous deposits in Argentina, dating to 70 to 135 Maya. The family Zamiaceae is more diverse, with a fossil record extending from the Middle Triassic to the Eocene, 54 to 200 Maya, in North and South America, Europe, Australia, and Antarctica, implying the family was present before the breakup of Pangaea. The family Cycadaceae is thought to be an early offshoot, with fossils from Eocene deposits, 38 to 54 Maya, in Japan, China, and North America, indicating this family originated in Laurasia. Cycas is the only genus in the family and contains 99 species, the most of any cycad genus. Molecular data have recently shown Cycas species in Australasia and the east coast of Africa are recent arrivals, suggesting adaptive radiation may have occurred. The current distribution of cycads may be due to radiations from a few ancestral types sequestered on Laurasia and Gondwana, or could be explained by genetic drift following the separation of already evolved genera. Both explanations account for the strict endemism across present continental lines. Distribution. Overall species diversity peaks at 17 degrees 15 N and 28 degrees 12 S, with a minor peak at the equator. There is therefore not a latitudinal diversity gradient towards the equator but towards the tropics. However, the peak in the northern tropics is largely due to Cycas in Asia and Zimi in the New World, whereas the peak in the southern tropics is due to Cycas again, and also to the diverse genus Encephalatos in southern and central Africa and Macrozemia in Australia. Thus, the distribution pattern of cycad species with latitude appears to be an artifact of the geographical isolation of cycad genera, and is dependent on the remaining species in each genus that did not follow the extinction pattern of their ancestors. Cycas is the only genus that has a broad geographical range and can thus be used to infer that cycads tend to live in the upper and lower tropics. This is probably because these areas have a drier climate with relatively cool winters. While cycads require some rainfall, they appear to be partly xerophytic. Conservation. In recent years, many cycads have been dwindling in numbers and may face risk of extinction because of theft and unscrupulous collection from their natural habitats, as well as from habitat destruction. Traffic. The Wildlife Monitoring Network are urging countries supplying the illegal trade in cycads to implement a blanket ban on all species to prevent further wild extinctions. About 23% of the 305 extant cycad species are either critically endangered or endangered, and 15% are vulnerable. Thus, 38% of cycads are on the IUCN Red List, 2004. And the other 62% are in the least concern and near threatened category, i.e., not actually on the red list, or are data deficient. This value has changed dramatically within the past few years. 46% of cycads were on the 1978 red list, and this rose to 82% in 1997. This was largely due to the recent discovery of over 150 new species, disagreements about classification, and uncertainty. This has not been helpful for conservation planning for the group. Zimi in the New World, Cycas in Asia and Encephalatos in Africa are the most threatened genera. This pattern reflects the pressures on species in these regions. At least 2 species, Encephalatos woodi and Encephalatos relictus, both from Africa, are confirmed extinct in the wild. Cycads are long-lived with infrequent reproduction, and most populations are small, putting them at risk of extinction from habitat destruction and stochastic environmental events. Regionally, Australian cycads are the least at risk, as they are locally common and habitat fragmentation is low. However, land management with fire is thought to be a threat to Australian species. African cycads are rare and are thought to be naturally decreasing due to small population sizes, and there is controversy over whether to let natural extinction processes act on these cycads. All cycads are in the CITES appendix, appearing under the heading Plant Kingdom and under 3 family names: Cycadaceae, Stangeriaceae and Zamiaceae. All cycads are CITES appendix 2 except the following. In Appendix I, Cycas bedomi, Stangeria areopus, all Ceratozamia, all Chigua, all Encephalatos, Microcycas calicoma. Cycad seeds from species on Appendix 2 are not CITES regulated. Appendix I seeds are treated the same as the plants. Uses. In Vanuatu, where the cycad is known by the Bislama name Namela, the tree has deep customary and spiritual significance. A single cycad leaf may be used as a taboo sign, while a pair of crossed cycad leaves is a peace sign and appears on the Vanuatu flag. The breaking off of fronds from a cycad leaf is used in traditional contexts as an aid to counting. Cycad leaves are used to decorate venues during festivals, marriages and other community celebrations. Horticulture. Cycads can be cut into pieces to make new plants, or by direct planting of the seeds. Propagation by seeds is the preferred method of growth, and two unique risks to their germination exist. One is that the seeds have no dormancy, so the embryo is biologically required to maintain growth in development, which means if the seed dries out, it dies. The second is that the emerging radicle and embryo can be very susceptible to fungal diseases in its early stages, when in unhygienic or excessively wet conditions. Thus, many cycad growers pre-germinate the seeds in moist, sterile media such as vermiculite or perlite. However, pre-germination is not necessary, and many report success by directly planting the seeds in regular potting soil. As with many plants, a combination of well-drained soil, sunlight, water and nutrients will help it to prosper. Although, because of their hardy nature, cycads do not necessarily require the most tender or careful treatment. They can grow in almost any medium, including soilless ones. One of the most common causes of cycad death is from rotting stems and roots due to overwatering. Some insects, particularly scale insects, some weevils and chewing insects can damage cycads. Though the pest is susceptible to insecticides such as the horticulture-soluble oil white oil, sometimes bacterial preparations may be used to control insect infestation on cycads. When some of the mature plants prepare for reproduction, though, the presence of weevils has been shown to help accomplish pollination. While the cycads have a reputation of slow growth, it is not always well-founded, and some actually grow quite fast, achieving reproductive maturity in 2 to 3 years, as with some Zamia species, while others in 15 years, as with some Cycas, Australian Macrozamia and Lepidozamia. Source of nutrition. The starch obtained from the stems of certain species is still used as food by some indigenous tribes. Tribal people also grind and suck the seeds to remove the nerve toxins that may be present, making the food source generally safe to eat, although often not all the toxin is removed. In addition, consumers of bush meat may face a health threat as the meat comes from game which may have eaten cycad seeds and carry traces of the toxin in body fat. Cycad, known as satetsu, Jap. Kanji. In Japanese, was traditionally a famine food in Okinawa, a last resort to turn to for sustenance during particularly difficult times. A period of particularly devastating poverty and famine in the 1920s, caused in large part by Japanese economic policies in the island prefecture, is known as Cycad hell or Satetsu Jigoku. Cycad meal, known as eenthu in Malayalam, is a common food in Kerala. Traditionally, the seeds were sliced and kept in direct sunlight or near the hearth during rainy season to promote drying. The drying process is carried out to reduce the toxin levels and as a means of preservation. The outer shell is subsequently removed and inner portion is ground into a flour. Properly dried cycad seed flour may be stored for several years without deterioration. Food items like pachu, eenthu kanji, eenthu payasam, etc., are made out of cycad seed powder. These food items are particularly prepared in heavy rainy seasons in Kerala. There is some indication that the regular consumption of starch derived from cycads is a factor in the development of lytico-bodic disease, a neurological disease with symptoms similar to those of Parkinson's disease and ALS. Lytico-bodic and its potential connection to cycadsin ingestion is one of the subjects explored in Oliver Sacks' 1997 book Island of the Colorblind. Cattle that graze in pastures containing cycads may ingest the leaves and seeds and develop the neurologic syndrome of cycad toxicosis known as Zimia staggers. See also Fossil Cycad National Monument, formerly in the U.S. state of South Dakota. Chamberlain, C.J. 1965, 1919. The Living Cycads. First ed. Reprinted. New York. Hafner. 172p. LCCN 65027079. Chaw. S. M. Parkinson. C. L. Cheng. Y. Vincent. T. M. Palmer. J. D. 2000. Seed plant phylogeny inferred from all three plant genomes. Monophyly of extant gymnosperms and origin of nettles from conifer. PDF. Proceedings of the National Academy of Sciences. 97. 8. 4086-4091. DOI. 10.1073 PNAS. 97.8.4086. PMC 18157. PMID 10,760,277. Chaw. S. M. Walters. T. W. Chang. C. C. Hu. S. H. Chen. S. H. 2005. A phylogeny of cycads. Cycadales. Inferred from chloroplast MATK gene. TRNK intron. And nuclear rDNA its region. Molecular phylogenetics and evolution. 37. 1. 214-234. DOI. 10.1016 J. YMPEV. 2005.01.006. Chaw. S. M. Zarkik. A. Sung. H. M. Liu. T. C. Lee. W. H. 1997. Molecular phylogeny of extant gymnosperms and seed plant evolution. Analysis of nuclear 18's rRNA sequences. Molecular biology and evolution. 14. 1. 56-68. DOI. 10.1093. Oxford Journals. Molbeth. A025702. PMID 9,754. Crape. W. L. 2000. Progress in Understanding Angiosperm History, Success, and Relationships. Darwin's Abominably Perplexing Phenomenon. Proceedings of the National Academy of Sciences. 97. 24. 12939-12941. DOI. 10.1073 PNAS 97.24.12939 PMC 34068 PMID 11,087,846 De Luca, Paolo. 1990. A Historical Perspective on Cycads from Antiquity to the Present. In Stevenson, D. ed. The Biology, Structure, and Systematics of the Cycadales. Memoirs of the New York Botanical Garden. 57. p. 1-7. ISBN 0-89327-350-3. Donaldson, J. S. 2003. Chapter 3. Regional Overview. Africa. In Donaldson, J. S. ed. Cycads. Status Survey and Conservation Action Plan. IUCN, SSC Cycad Specialist Group. Gland, Cambridge, UK. IUCN. ISBN 2-8317-06998. p. 9-19. Donaldson, J. 2004. Saving Ghosts. The Implications of Taxonomic Uncertainty and Shifting Infrageneric Concepts in the Cycadales for Red Listing and Conservation Planning. In Walters, T. and Osborne, R. eds. Cycad Classification, Concepts and Recommendations. Wallingford, UK. CABI. ISBN 0-85199-741-4. p. 13-22. Donaldson, J. Hill, K. D. and Stevenson, D. W. 2003a. Chapter 2. Cycads of the World. An Overview. In Donaldson, J. S. ed. Cycads. Status Survey and Conservation Action Plan. IUCN, SSC Cycad Specialist Group. Gland, Cambridge, UK. IUCN. ISBN 2-8317-06998. p. 328. Donaldson, J. S. Dehagen, A. P. Vovides, A. P. and Tang, W. 2003b. Chapter 7. Cycads in Trade and Sustainable Use of Cycad Populations. In Donaldson, J. S. ed. Cycads. Status Survey and Conservation Action Plan. IUCN, SSC Cycad Specialist Group. Gland, Cambridge, UK. IUCN. ISBN 2-8317-06998. p. 39-47. Golding, J. S. Herter, P. J. H. 2003. A Red List Account of Africa's Cycads and Implications of Considering Life History and Threats. Biodiversity and Conservation. 12. 3. 507-528. DOI 10.1023a 1,422,801,638. Gonzalez Astorga, J. Vovides, A. P. Ferrer, M. M. Iglesias, C. 2003a. Population Genetics of Dioecious Lindell. Zamiaceae, Cycadales. Biogeographical and Evolutionary Implications. Botanical Journal of the Linnean Society. 80. 3. 457-467. DOI 10.1046 J. 1095-28312.2003.00257. X. Gonzalez Astorga, J. Vovides, A. P. Iglesias, C. 2003b. Morphological and Geographic Variation in Dioecious Lindell. Zamiaceae. Ecological and Evolutionary Implications. Botanical Journal of the Linnean Society. 141. 4. 465 to 470. DOI 10.1046 J 1095 to 8339.2003.00155 X Gregory T J Inkemnik J 2004. Hypotheses of the relationship between biogeography and speciation in Dion Zamiaceae in Walters T and Osborne R eds. Cycad classification, concepts and recommendations. Wallingford, UK. CABI. ISBN 0-85199-741-4. p. 137 to 148. Hermson, E. Taylor, E. Taylor, T. Stevenson, D. 2006. Catafalls of the Middle Triassic Cycad Antarctocycas Shofi and New Insights into Cycad Evolution. American Journal of Botany. 93. 724 to 738. 2006. DOI 10.3732 AJB 93.5.724. PMID 21642136. Hill, C. R. 1990. A ultrastructure of in situ fossil cycad pollen from the English Jurassic, with a description of the male cone Androstrobus barme sp. November. Review of Paleobotany and Palynology. 65. 1-4. 165 to 193. DOI 10.1016 0034 6667 90 90067S. Hill, K. D. 2003. Chapter 4. Regional Overview. Australia. In. Donaldson, J. S. ed. Cycads. Status Survey and Conservation Action Plan. IUCN, SSC Cycad Specialist Group. Gland, Cambridge, UK. IUCN. ISBN 2-8317-0699-8. p. 20-24. Hill, K. D. 2004. Character Evolution. Species Recognition and Classification Concepts in the Cycadaceae. In. Walters, T. and Osborne, R. eds. Cycad Classification, Concepts and Recommendations. Wallingford, UK. CABI. ISBN 0-85199-741-4. p. 23-44. CABI. Oxford. Hill, K. and Stevenson, D. 1998 present. The Cycad Pages. http. PlantNet. RBGSYD. NSW. Gov. O. PlantNet. Cycad. Hill, K. D. Stevenson, D. W. Obsorn, R. 2004. The World List of Cycads. Botanical Review. 70. 2. 274 to 298. DOI 10.1663 0006 8101. 2004. 070 0274. TWLOC 2.0 CO 2. Hopkins, DJ. Johnson, K.R. First record of cycad leaves from the Eocene Republic Flora. Washington Geology. 25. 4. 37. Jones, David L. 1993. Cycads of the World. Washington, D. C. Smithsonian Institution Press. ISBN 1-56098-220-9. Second ed. Republished. 2002. As. Cycads of the World. Ancient Plants in Today's Landscape. Sydney. Reed New Holland. ISBN 1-876334-69-X. Keppel, G. Lee, S. W. Hotchkiss, P. D. 2002. Evidence for long isolation among populations of a Pacific cycad. Genetic diversity and differentiation in Cycas Semeneia. Br. Cycadaceae. Quote. Journal of Heredity. 93. 2. 133–139. DOI. 10.1093. Jade. 93.2.133. Norstog. K. J. and Nichols. T. J. 1998. Biology of Cycads. Ithaca. Cornell University Press. ISBN 0-8014-3033-X. Stevenson. D. 1992. A Formal Classification of the Extant Cycads. Britonia. 44. 220–223. DOI. 10.200307 Stevenson. D. K. Norstog. and P. Fawcett. 1998. Pollination Biology of Cycads. In Reproductive Biology. In Systematics, Conservation, and Economic Botany. eds. S. Owens and P. Rudel. pp. 277–294. Royal Botanic Gardens. Q. Walters. T. Osborne. R. and Decker. D. 2004. We hold these truths. In Walters. T. and Osborne. R. eds. Cycad Classification. Concepts and Recommendations. Wallingford, UK. CABI. ISBN 0-85199-741-4. p. 1–11. Whitelock, L. M. 2002. The Cycads. Portland. Or. Timber Press. ISBN 0-88192-522-5. Palm trees. Small palms. Cycads. Bromeliads and tropical plants site with thousands of large, high-quality photos of cycads and associated flora. Includes information on habitat and cultivation. Hill K.D. 1998–2004. The Cycad Pages. Royal Botanic Gardens, Sydney. http. PlantNet. RBGSYD. NSW. Gov. O. PlantNet. Cycad Index. HTML. Gymnosperm Database. Cycads. Fairchild Tropical Botanic Garden, one of the largest collection of cycads in the world, in Florida. U.S.A. Palm and Cycad Societies of Australia. PACSOA. The Cycad Society of South Africa. Cycad nitrogen fixation. Cycad toxicity. Cycads. Photo. The Cult of the Cycads. New York Times Magazine article on Cycad collectorship and Cycad smuggling. Cycads, an annotated link directory.